Anika Retzmann, Universität für Bodenkultur Wien, Austria.
Where are you from? That's one of the questions anthropologists and archaeologists ask themselves when they find remains. We as analytical chemists can help to answer this question by using, for example, strontium isotopic analysis. As the individual lives in a distinct geographic area, it incorporates the chemical and isotopic fingerprint of its environment by food and water into bone and tooth material. If this individual moves to another place during the lifetime and dies there, the surrounding soil has a different isotopic fingerprint than those ones, for example, stored from the childhood in tooth and bone material. While stored, it might happen that ions from the environment move into the bone and overprint the original isotopic fingerprint in the bone material. That's called diagenesis. That's a problem, because we can't see it with the naked eye. And our idea here is to use hyperspectral imaging as a cheaper and faster way to monitor these diagenetically changed regions on bones. In hyperspectral imaging, we are taking pictures over the whole visible and near-red light spectra. Means for 1 pixel of our picture, we're getting somewhere around 250 spectral absorption band informations. That's a whole bunch of data. We're going to process these data with multivariate image analysis by plotting those pixels with similar information. By identifying those spectral information of diagenesis, going back to the bone, we are able to monitor, to highlight these regions which are diagenetically changed and those ones which are still preserving the original biogenetic information. By taking samples from these biogenetic regions on the bone and doing, for example, strontium isotopic analysis with mass spectrometry, we are going to be able to determine whether this individual is local or not. Sounds interesting for you? Might be thinking, yeah, cool for archaeology, but what's in it for us? My answer to that is, think about it. Imagine what we can do in quality control and provenancing if we apply this technique, let me say, for example, on food or wood or every other thing you can naturally grow, if we just apply the technique on the right spot, which is uncontaminated. Thank you.